We'll start. Good morning or good afternoon, wherever you are. Welcome to our last webinar of 2024, but the first in our new building, in our new headquarters with new production facilities. So it's a new start for us. So today we'll present to you our brain organoid-on-chip model, and we'll focus specifically on neurotoxicity applications. So today we'll have two speakers. We will have Eloise Castiglione, who is our PhD student that works in collaboration with NATRI and Subbiotech CA Joint Laboratory. So her main PhD project is on development of a brain organoid-on-chip platform. And we have Jessica Rontard, that maybe some of you have already met in our webinar sessions. She's a biopharma scientist with an extensive expertise in neurosciences, human stem cells, and neurodegenerative disorders. And today she's leading at NATRI the R&D of CNS platforms. So first, I will give you a quick explanation of what we do at NATRI, and then we will get into our brain organoid-on-chip model and its improved reproducibility compared to conventional supports. Of course, we will then explain how this model can be used in neurotoxicity studies, and as usually, we will end with a Q&A session. So do not hesitate if you have any questions during the webinar to put them in the chat box, and we will, at the end of the webinar, we'll answer all of them. So, as many of you know, today high-throughput solutions for drug screening, for example, usually they're based on 96 or 384 well plates, or even 24 well plates for example, organoids. And so in these wells, usually when you want to study complex systems, you would usually put different or mixed cell types in contact, such as, for example, neurons and a target cell or target organ. For example, you can put in contact, for example, keratinocytes with neurons. However, this is not representative of human biology, where we know that there is a physiological compartmentalization. A first one with the skin being the interface between external environment and internal environment, but you also have internally different compartmentalization with the nervous system, for example, the vascular system and the different organs. And so that's where the microfluidic solutions and what we do at NATRI comes into play. We do, as you see on the right, you have an example of a microfluidic chip with the individual one, which is from our dual link architecture, where you have 3 channels, with the middle channel being much thinner than the 2 on the left and on the right channel. And this middle channel, it mainly has a focus on the action of creating a microfluidic barrier. And so in those systems, you can actually culture, for example, on the scheme in the middle, you have one cell type, for example, pulmonary cells. You can seed them and culture them in the first channel. And then you can culture, for example, sensory neurons on the third channel. And so during the culture, you will have neuronal endings, axons, that will grow, pass through all of the channels, through microchannels, and then it will go and innervate your target organ or target cell. And that way, compared to cultures in wells, you do have a compartmentalization where you have your target cells that will be in contact only with nervous endings, and your somas will stay in a separate compartment. And so these chips, they come in a 96 well plate format. And so this system works for 2D cultures. However, we have launched recently an early version of a chip that is specific for 3D cultures. It's what you see in the middle down below, which is a system with a membrane. So you have a middle well in yellow, where you can harbor and culture, for example, a brain organoid or other organoids, in fact. On the bottom, you have a perfusion reservoir, where you can perfuse different drugs, or even you can culture endothelial cells and that way you can mimic the vascularization of your organoid. So, of course, our chips, they are, as you can see, they are transparent. So they're made from a breathable silicone that is permeable to oxygen. And that way, you can maintain your cultures without pumps or additional devices. And because the silicone is transparent, you can also use it for example, imaging. And in this case, what we work today at Natri, what we have done is a step forward, and we added MEA, microelectrode arrays, underneath some of our chips. And that way, as we know, neurons, they can vehiculate through action potential different electrophysiological signals. And so the idea is that because neurons, they interconnect different organs, and so they vehiculate different types of messages depending if your organ is healthy, or pathological, or if you have different types of stimuli. These are messages that are encoded as electrophysiological signal and vehiculated by neurons. And so the idea is to capture these electrophysiological signals to microelectrode arrays, and then construct a digital library with reference compounds or healthy types of cells versus potentially pathological cells, and be able to create these predictive models to be able to predict, for example, the efficacy or the toxicity. And overall, so you can, in fact, we do produce our devices for the platforms. So we can sell those to our clients. But of course, we also have, because we have our own R&D biology laboratories, so we also can perform screening services or even co-develop new models. And these models, they can range from, for example, sensory, innervated skin, and of course, brain organoids is what the topic of today's webinar is. Of course, when we work on different projects, we can work on different levels of complexity, with the basic one being studying effects of different compounds, or for example, neurons alone. We can also study for example, supernatant of cells, if we want to test different metabolites or different cell secretions on the neurons, and the complex one being to have co-cultures. So, of course, today our subject is brain organoids. For brain organoids, we have used our duplex well device, as I mentioned previously. So you have an image of what the duplex device chip could look like on the right. And in the middle, you have a schematic where your organoid will be. So you can harbor and culture a brain organoid in the central well in yellow. It is separated from the bottom perfusion channel by a membrane that is porous. And so in the perfusion chamber, you can test different drugs, but of course you can also test drugs directly in the open well. And you can also create a vascular system underneath your organoid by culturing, for example, endothelial cells. And so these devices, they come in two versions. You have a version that is a plate with 8 chips, and you have a full version with 16 chips. And so now I will let Eloise take the lead.
Okay, thank you, Nasiba. So now let's move on with the presentation of our brain organoid-on-chip model. And first, to recontextualize, cerebral organoids are 3D self-organized cellular structures obtained in vitro by neuroectodermal differentiation of pluripotent stem cells, and they comprise different cell types observed in the human embryonic brain, including neural progenitors, neurons, and astrocytes. And these cell types are able to self-organize in 3D and to form cytoarchitectures that are similar to certain regions of the human developing brain. And so, as such, cerebral organoids have become relevant in vitro models to study the human brain development and functioning, and may become promising tools for neurotoxicity evaluations and drug screening in preclinical studies. However, one of their main limitations is the high heterogeneity observed between cerebral organoids, including differences of morphology and size, but also structural and functional discrepancies. And this induces a lack of reproducibility and predictability of cerebral organoids, which currently prevents their industrial transfer. So, facing these limitations, a critical issue is to find a solution to enhance the reproducibility of cerebral organoids. And in the literature, some publications have underlined the role that microfluidic devices could play. So microfluidic devices offer several advantages compared to conventional culture supports. Thanks to tightly controlled fluid flows, they enable to recapitulate in a more physiologically relevant manner the cellular microenvironment, which is considered to globally improve the culture conditions and the reproducibility between the cell cultures. And as Nasiba mentioned, Netri develops microfluidic devices that are standardized and compatible with industrial formats and readouts. So in the light of this, our objective is to combine cerebral organoid culture in a Netri's microfluidic device, and more specifically in the duplex device that was presented before, to develop a brain organoid-on-chip model to improve the culture conditions and the reproducibility of cerebral organoids with a perspective of facilitating their industrial transfer for preclinical applications. So first, we will start with a presentation of this brain organoid-on-chip model, the optimization of the on-chip culture conditions, and results demonstrating an improved reproducibility on-chip compared with conventional culture supports. So regarding the cerebral organoid culture protocol, we start from iPSCs that we force to aggregate to form embryoid bodies, EBs. The EBs are then harvested and cultured in successive culture media that contain factors that will induce the neural differentiation in 3D. And more specifically, we apply a regionalized differentiation protocol that guides the differentiation towards the cortical region of the brain, leading to cortical organoids. And so, in order to optimize the cortical organoids culture conditions on chip in the duplex device, we assayed several conditions. First, we tested to introduce the organoids in the device at several time points. The time point D2, which is the earliest time point where the EBs have just formed, and also the time points D11 and D18, which are strategic time points in the early cortical differentiation and associated with changes in the culture media compositions. And given the architecture of the duplex, it is possible to renew the culture medium independently in the top chamber and the bottom channel. So we tested 3 methods of medium renewal. A first method where the medium was changed by the bottom and by the top every 2 days. A second method where the medium was changed by the bottom every 2 days and only once per week by the top. And a third method where the medium was changed only via the bottom channel. And so the first cultures demonstrated that the on-chip organoids were similar to control organoids cultured in 24 well plates, the conventional culture support, and that they displayed the expected morphology, cell types, cytoarchitectures, and RNA expression levels after 60 and 120 days, so after 2 and 4 months of culture. And regarding the on-chip protocol, the first condition of medium renewal, so by the top and the bottom, seemed more appropriate since the 2 other methods with reduced changes by the top could lead in some cases to organoid adherence onto the membrane. And the 2 latest time points, D11 and D18, seem more suitable than D2 with organoids of better quality at the end of culture. And in addition to the optimization of these on-chip culture conditions, these first results also demonstrated a tendency towards an improved reproducibility on-chip compared with conventional culture support, especially regarding the organoid sizes. So by plotting the organoid size dispersions in green for control organoids cultured in 24 well plates and in blue for organoids cultured on-chip, we can observe that the on-chip organoid display sizes that are smaller on average, but much more homogeneous compared to the controls in 24 well plates, and this during all the culture up to 60 days. And similarly, We also observe this pattern with sizes that are smaller on average, but much more homogeneous for the on-chip organoids at a more mature time point of 120 days, here represented in pink on the second graph. And we have also demonstrated a tendency towards an improved reproducibility on chip of the cytoarchitectural organization after 60 days of culture. So on this immunofluorescence staining, we can observe that the on-chip organoids display the expected cell types and cytoarchitectures with the presence of neural progenitors in red that form rosettes, which are neurogenic areas modeling the neural tube. And we can see that these rosettes seem better organized and in a more reproducible manner for the on-chip organoids, where they are always small, round-shaped, and present only on the organoid borders, while for the control in 24-well plates, these rosettes seem less organized, with some organoids displaying very few rosettes randomly distributed, and some organoids displaying rosettes that are more or less organized on the borders. So globally, the cytoarchitectural organization seems improved, of better quality, and more reproducible for the on-chip organoids. And similarly, we also observed this improvement of the cytoarchitectural organization and this gain of reproducibility at a more mature time point of 120 days. So at this time point, we can see that the on-chip organoids display the expected markers, especially with the presence of cortical neurons belonging to the upper and deep cortical layers in red and green. And we can observe that these cortical layers are well formed and present only on the borders of the organoids for all the on-chip organoids, while for the controls in 24-well plates, these cortical neurons seem less organized and do not always form a distinct cortical layer as represented here. So, taken together, these results suggest a tendency towards an improved reproducibility on chip in terms of organoid sizes and global cytoarchitectural organization, as well as an improvement of the quality of these cytoarchitectural organizations. And so, in order to better characterize the cortical organoids and to evaluate their quality, we have developed a quality control system adapted to 60-day cortical organoids. So it relies on several criteria for organoid analysis. First, the organoid morphology, then the size and growth profile, the cellular populations, the cytoarchitectures, and the cytotoxicity level. And for each index, a score is attributed to the evaluated organoid, ranging from 5 for good quality to 0 for low quality, and minimal scores to pass the quality control have been defined for each index. So for a cortical organoid to pass this quality control, it has to reach sufficient scores for each index, otherwise it fails and should be excluded from the given study. So by permitting an objective and reproducible quality assessment, this quality control opens the way for improved consistency and comparability of results in the field of cerebral organoids. So we have seen so far that our brain organoid-on-chip model tends to improve the quality and the reproducibility of cortical organoids, and we have developed a quality control system for facilitating the quality assessment of these cortical organoids. Therefore, our brain organoid-on-chip platform associated with the quality control opened the way for preclinical applications, such as neurotoxicity studies. As a proof of concept, we performed preliminary neurotoxicity studies using this model. So briefly, we tested two compounds, the vanillin and the biphenyl, in acute and chronic exposures at several different doses. And we have put here an example of results for an acute exposure with the highest dose of vanillin, the first compound, and we can observe that the exposed organoid displayed an optimal morphology and was similar to the non-exposed control and exhibited the expected cell types and cytoarchitectures after 60 days of culture, suggesting an absence of toxicity induced by this dose of vanillin. However, we have put also another example of an acute exposure with the highest dose of biphenyl, the second compound, and here we can observe that the exposed organoid displayed an altered morphology with the presence of a large cyst, disorganized cytoarchitectures, and higher apoptosis and DNA damage levels compared to the non-exposed controls, suggesting a toxicity induced by this dose of biphenyl. And so, taken together, the results of these neurotoxicity studies demonstrated no discernible impact of the vanillin on the neurodevelopment in cortical organoids, while they revealed a toxicity induced by the biphenyl in a dose-response manner. And so finally, within the framework of these neurotoxicity studies, in order to better characterize the cortical organoids exposed to a compound and to be able to classify the assayed compound in terms of neurotoxicity level, we have developed what we call a prediction algorithm that relies again on several criteria for organoid analysis and a scoring system, similarly to the quality control that we have presented before, but here tailored specifically for toxicity evaluations. And so depending on the scores obtained for each index, it will guide the decision process towards one of the three classification categories for the assayed compound: high toxicity, potential toxicity, or low toxicity. In conclusion, our brain organoid-on-chip model enables the culture of cortical organoids with several possible on-chip culture conditions. We demonstrated an improved reproducibility on-chip in terms of organoid sizes and global cytoarchitectural organization. We also developed a quality control system for facilitating the quality assessment of these cortical organoids. And we have applied the brain organoid-on-chip model for neurotoxicity studies, along with the development of a prediction algorithm to help compound classification. And so together, the brain organoid-on-chip model used for neurotoxicity studies associated with the prediction algorithm opened the way for compound screening for neurotoxicity evaluations in fields such as drug development, chemical risk assessment, food safety, ecotoxicology, etc. And so in perspectives, we work towards implementing an electrophysiological activity recording of the organoids directly on chip. And we also aim at complexifying this model with the addition of a blood-brain barrier modeling and to further continue the neurotoxicity studies using these models.
Yeah, so thank you, Eloise, and thank you, everyone. Hopefully, everything was clear. So we'll start our Q&A session, so do not hesitate to share any questions that you have in the chat box. And for the Q&A session, of course, we will be joined by Jessica. So a first question, how many organoids do you put in each chip? So it's probably more for Jessica.
Yes, thank you for this question. We culture one cerebral organoid per duplex device to follow them individually and to prevent the organoid from fusing. Just to note, using the neobento format that we have presented before, it is possible to work with 16 devices per plate, so 16 individual cerebral organoids per plate. The dimensions of the open well in the duplex are 5 per 5 per 6 millimeters, make it also possible to maintain several organoid depending on the size and the specific application. So please feel free to contact us for more information.
Thank you, Jessica. Another question for Eloise. Have you tested other cell lines?
Yes, we have tested 2 IPS lines. So most of the experiments that were presented are based on one IPS line from an healthy donor, but we have also tested another IPS line from an healthy donor. and up to 60 days of culture, and we observed similar results in terms of sizes and cytoarchitectures with the two lines.
Okay, thank you. Another question for Eloise. You demonstrated the use of this platform for neurotoxicity assays, but could also it be used for therapeutic screening with pathological organoids? Are there any changes to make to do so?
Yes, it could be completely suitable also for pathological modeling. We just have to take into account in the quality control system the specificities and the hallmarks of the studied pathology, and also to adapt the prediction algorithm because the outcomes will be dependent on the specific hallmarks and phenotypes, but it will be completely compatible with the brain organoid-on-chip model.
Okay, thanks. Hopefully we answered your question. Another one, are your cerebral organoid cultured under agitation? So Eloise, I think you can answer that.
Yes, exactly like for the conventional culture conditions in 24 well plates that we put under constant agitation, we also put the chips under agitation using an orbital shaker at a speed of 18 RPM. Otherwise, we have noticed that the organoids tend to adhere onto the membrane of the duplex chip without agitation.
Okay, thanks. We have another question on how we work on our organoids. Do you use Matrigel for your organoids?
Yes, thank you for this question. So just to remind, Matrigel is an hydrogel that is an equivalent of extracellular matrix and serves as a 3D scaffold for the organoids. But there are some animal compounds and it's not well defined with some variation from batch to batch. So it's not really compatible for preclinical application. For these reasons, we follow a protocol in which there is no need of Matrigel. As Eloise presented before, we used a regionalized differentiation protocol for cortical organoids. But we have also tested the unguided protocol from Lancaster and collaborators with a Matrigel embedding of the organoid. So yes, the duplex devices is compatible with the use of hydrogel.
Okay, thanks. Are there any other questions? Someone is interested to know if it's possible to record electrophysiological activity on the brain organoids in our chips.
Yes, I can take this question. It's not possible to record the electrophysiological activity directly in the duplex device. However, we are developing a new devices called Penthouse, adapted for 3D cell culture and combined with electrodes to perform MEA recording and follow the electrophysiological activity in the cerebral organoids. So please free to contact us for more information.
Thanks, Jessica. Okay, so we have several other questions. What is the difference in media consumption in 24 well plate and on-chip platforms? So maybe Eloise, as you know best.
Yes, it's a very interesting question because obviously in the chip, the volume is very smaller compared to the 24 well plates, where there is only 150 microliter in the top chamber and 150 microliter in the channel. While in the 24 well plates, we put 1 ml. Yes, 1 ml per well. So that's for the reason why we have to renew the medium in the chip every 2 days. Otherwise, the medium consumption is too quick and the medium becomes yellow. So we have to renew it more often compared to the 24 well plates.
Okay. Another question. Great work. Thank you. At the end, you mentioned about including blood-brain barrier together with the neural organoids. Could you please elaborate on that? Which type of cells will you use for blood-brain barrier model?
Yes, thank you for this question. We imagine to add 3 other cell types, so astrocytes, pericytes, and also brain endothelial cells on the bottom of the device. On the bottom, yes, sorry.
So did we answer your question, hopefully? Are there any more questions? Okay, so we have another question, I think. Okay, so we have another question. Regarding your organoids in the chip, is there any improvements regarding apoptosis necrotic core or this phenomenon of vesiculation or cyst formation? Go ahead, Eloise.
Yes, it's a very interesting question. Regarding the necrotic core, in fact, we don't really have this issue, even in the conventional cultures, probably because up to 2 and 4 months of culture is not so long and the organoids do not grow so much. So we do not have this problem, so we can't really compare. But for the vesiculation part, it seems to be reduced in the chip. We have much less this problem compared to the 24 well plates, where they tend to often have this cyst forming in every organoid batches and much less in the chips.
Thank you, Eloise. So do you have any more questions? Okay, one last question. So what is the possibility of using on-chip for co-culture experiments such as tumor invasion?
Yes, maybe I can answer. I think it would be completely possible with the duplex architecture, with the 2 compartments, to have the tumor invasion in the bottom channel on the membrane if the organoid above in the culture chamber adheres onto the membrane and there is kind of direct contact. Otherwise, it would be possible also to make a co-culture of, for instance, glioblastoma and cerebral organoids. It is often done and already been done by some labs. And yes, so several possibilities.
Okay, that's interesting. Thank you, Eloise. Do you have any last questions? Okay, so in case we haven't had the time to reply to your questions or you have any questions afterwards, do not hesitate to contact us. You have our email address, contact@netri.com, and do not hesitate to even rewatch some of our older webinars. And I hope, I wish you a happy New Year in advance, and see you next time. Thank you. Bye.